Hi everybody. Welcome to part 3 of the bioinformatics lectures for the next generation sequencing module here in UWC Biotechnology. I'm really glad to see you all today. For people who are joining us online, look at the description below this video. You'll find the link to the slides. That way you don't have to worry about reading it off of this fuzzy screen behind me. So we are going to start today with the discussion of gene expression. Gene expression is a hugely important task, and it's going to take us through bioinformatics into the fine world of biostatistics as well. This is a field that we all need a little bit of background in. I want to note that the first few times I taught this subject, I found that I didn't understand a lot of aspects of microarrays well enough. And I borrowed a lot of slides from Bing Zhang, a friend of mine, formerly at Vanderbilt University, now at Baylor College of Medicine. So I always appreciate him whenever I get to see these slides again. We're going to be talking about the biotechnology of gene expression via microarrays, yes, but also via RNA Seq. Does everyone know the distinction between microarray and RNA Seq expression? Oh, okay, well we'll cover some of that. One is a technique of hybridization of probe sequences on a physical surface with bits of cDNA, and the other one is a sequencing exercise. So we'll talk about the purpose of these, the technology that makes them possible, and have a whole lot on the data analysis that makes this interpretation feasible. And from there, we will talk a little bit more about high-throughput sequencing and how it plays into this. There's plenty of talk about statistics as well that feeds into this lecture. Most of that will come at the end. Okay. Now, I remind you that all of what we've been talking about in these 3 lectures fits somewhere on this chart, really. So we're going to work our way down this green fork at the right as we talk about this material. But you remember our discussion of assembly and annotation. That was the first day. We had amplicon sequencing, which doesn't actually feel represented on this slide just yet. I need to probably update that in further years. But today we're going to be talking about what we can accomplish through RNA Seq and transcript microarrays. Okay, so why is it that we need to measure gene expression? What is the difference between the genome in a neuron and the genome in that same person's liver? What's that?
Levels of expression.
Levels of expression. All right, so what sequences we have in the genome don't differ at all. It is possible for a person to contain multiple genomes, though. I mean, what if you had a tumor, for example, right? That tumor might have sequence variants throughout it. But generally speaking, when we talk about what genes are present in that individual, once you know the catalog, and there's really nothing else to know. So gene expression is one of these things that we need to talk about as why a liver cell and a nerve cell have such different physiologies. So it's highly regulated in mutant cells versus wild type cells. I already mentioned an example there. You might have a tumor, and in that tumor you might have heterogeneity. You might have some cells that are very adventurous and trying to spam the rest of the body with themselves, whereas wild type cells grow until they meet their neighbors and then they stop. All right. You may have certain stimuli that alter gene expression. You might have drugs that are impacting that. You might have a light signal. You might have sleep going on, and all of these things can change the pattern of expression for that. You might have different developmental stages. Certainly you have some genes that are only active when you're an embryo or at a later stage, at a fetal stage. And those genes may be completely quiet now that you're a grown-up. So that's also the case. You may have different cell types. If you have a bit of meat you've taken from somebody through a biopsy, you may see that even in a relatively small pitch of material, you have different kinds of cells at different layers within that. And certainly, diseased cells may behave quite differently than those that are healthy. If you have a macrophage, for example, that's gobbled up Mycobacterium tuberculosis, its behavior is going to change as a result of that. So we typically talk about the number of messenger RNA copies that we have in the cell for a given gene as an indicator of its corresponding protein expression level. How we measure that may be something that produces counts, or it may be something that produces a light signal for us to measure, depending on whether you're talking about sequencing or arrays. I always find that it's useful to review the structure of genes, just to remind people why expression can vary. So let's start with the idea, let's start with prokaryotic organisms. Does anyone want to name one for me? E. coli, thank you, all right. E. coli, our old friend, about 4,400 genes as I recall. Some of them are organized into what we call a polycistronic operon. So in this case, we have an enhancer region, an operator, a promoter. Maybe this is one of these Tata boxes. Here we have the untranslated region. We have a region of start to stop right here within the genome. And then we have yet another start to stop region with another UTR packed in between them. So in this case, we have one operon that is going to produce multiple messenger RNAs. You see this? So we get out a long RNA, and from that, we see that there are multiple RBSs here. What's the RBS? Anyone remember? Ribosomal binding site, exactly. These are sites where a ribosome sits down on this RNA and says, this is a great place to start translating. And boom, off comes this polypeptide from this first open reading frame. And here, if the ribosome instead latches onto this point, we get a translation of this region to produce this protein. So we have one region that produces an RNA with multiple ribosomal binding sites, and thus we get multiple polypeptides produced from it. That's quite different than what we get in the eukaryotic world. So we still have pieces like promoters and untranslated regions and enhancers and silencers, but when we get to the open reading frame, we see that it dissects into two different kinds of blocks. We have exons, and we have introns. Introns are parts that can get spliced out, whereas the exons get stitched together to produce one single messenger RNA. Now, although this doesn't really view it, doesn't visualize it, imagine that only the first intron is spliced out. In a case, sorry, I'm not making a lot of sense. Let us imagine that this point of this pre-mRNA gets glommed onto this part. Only the first and last exon would be merged together to produce a messenger RNA. If both of these pieces get spliced out, we instead have all three of these exons represented in a messenger RNA. So this process of RNA splicing can lead to multiple transcripts being possible for a single gene. Everyone gets that? Great. So maybe instead of 22,000 genes in a human, you may have something like 70,000 different transcripts. Maybe that number is higher. But I think 70,000 is at least a minimum number on how many different transcripts our genomes can produce. So any one of those messenger RNAs may then be translated, again by a ribosome, slightly different structure in the eukaryotes than in prokaryotes, but you get a protein out for that particular transcript. So even though different proteins may come from the same gene, if they've come from different transcripts, they will have different sequences. Okay, on we go. How do we measure that? So we're going to talk then about the set of all messenger RNA molecules produced by a cell as a transcriptome. Transcriptome. That is not a term that gets used a lot. Of all the omics, we hear an awful lot about genomics, an awful lot about proteomics, an awful lot about metabolomics for that matter, but you don't frequently hear people talk about transcriptomics anymore. That's kind of an odd feature of this. You'll sometimes see it masquerading under the name functional genomics instead, but just the same, you're talking about gene expression. So we have hybridization-based approaches and sequencing-based approaches. So you can label that in your mind, that second bullet point, hybridization-based approaches. I'm generally going to be talking about microarrays when we talk about hybridization-based approaches. Sequencing-based approaches are things more like RNA sequencing, RNA Seq as we call it. So since this is the next-gen sequencing module, you can expect that we'll be placing an emphasis on the latter rather than the former. But we'll be talking about some of the perks of each of these along the way. All right, so now why do we spend so much time talking about DNA microarrays and DNA sequencing in RNA Seq experiment as what we're doing? The answer is that messenger RNAs kind of get twitchy. If you look at them funny, they dissolve, right? They go away, they get digested up by something. So messenger RNAs are really kind of shady customers, and we want them to be nice and stable for us to measure them. So one of the first things that you frequently find we do is to produce complementary DNA, cDNA, from it. Is this familiar to everybody already? Okay, great. I'm glad you probably have much more experience in the lab of doing these things than I do. So you may produce a complementary DNA copy in order to have a molecule that's more stable for you to measure later. And you may use that opportunity to amplify it up as well, to amplify the signal of what you're working with. So cDNA is one of these, is kind of our more stable copy for us to do our measurements. All right. How do we make microarrays? There are two different approaches that I'm going to spell out here, and I think that my next slide actually talks about this latter approach a bit more, so I'm going to hold off on that for a moment. We speak of microarrays as a Cartesian array of DNA probe sequences. Has anyone heard the word Cartesian before? Good, okay. Where does the word Cartesian come from?
Cartesian plane,
Good. Yes, very nice. Okay, a Cartesian plane is a two-dimensional structure that we use in a lot of our old geometry classes. Does anyone know who it's named after?
Mr. Cartesian.
Mr. Cartesian. Good guess, but wrong, actually. René Descartes. René Descartes is known as a philosopher, but he was also quite the mathematician. And he was one of these first folks to say, you should put an x-axis along this, on a horizontal line, and a corresponding y-axis along a vertical line, and from that we can plot functions of x and y. So we call that a Cartesian plane in honor of René Descartes. Okay, so these arrays then are a whole bunch of x locations and a whole bunch of y locations, a bunch of rows and a bunch of columns, essentially. And each of these little locations, each of these grid points that are formed by that Cartesian array, is going to have a whole bunch of probe sequences. The probe sequences at this particular location all correspond to a particular messenger RNA we want to measure, or a particular cDNA that we want to measure. All right, so if you have 100,000 x and y coordinates on these plots, you would have 100,000 different transcripts that were getting measured, unless you had some redundancy built into it. You might imagine that you have each gene represented in two different locations on the array to help you evaluate which probes you could trust and which ones you couldn't. So this spotted or printed array represents a case where we're using something like an inkjet technology. Does anyone remember inkjet printers? You guys ever have an inkjet? Okay, a few folks did. Inkjet printers are able to deposit particular dyes, particular inks, onto a piece of paper with great precision, maybe down to 720 dots per inch, right? That's a vanishingly small dot. So imagine that instead of spraying ink, your printer is instead spraying little bits of DNA onto the surface, right? And the surface is set up so it will adhere to the DNA you spray at each of these locations on this glass slide, basically. Okay, so that is the spotted or printed array. And you saw in the early days, Agilent, GE, and ABI all making use of this technology for creating these arrays. It's great if you have a customized set of DNA sequences that you want to deposit onto this probe surface. It's also a relatively inexpensive way to pursue the creation of a microarray. However, spotted or printed arrays tend to have fewer features on them. They tend to have fewer different probes that you can load onto one surface. So that's kind of a weakness of them. As a result, we saw an awful lot of people move in the direction of synthesized arrays. Now, synthesized arrays have this really odd thing, and you probably just won't believe me when I tell you at first. We synthesize the DNA right on the surface itself. We're building DNA chains, the probe sequences, right on the surface. So how do we do that? Again, I'm going to use an example from computing technology. Has anyone heard of photolithography? No one has, okay. Does anyone have a photolithographic device with you today? The answer is everybody does. Photolithography is the process by which we manufacture CPUs. And we have the ability to print transistors that are, I think the current standard is something like 12 microns in size. Vanishingly small. So we are able to synthesize very small features using a technique called masking. So I want you to imagine that I have some surface on which I want to grow a whole bunch of different DNA sequences. And remember, I want a whole bunch of copies at this location for a particular sequence. I have a whole bunch of copies for a completely different sequence over here. So imagine that I start by masking this whole surface. Basically, I'm masking this surface such that at no place is it possible to add a nucleotide. So I've masked. Next, I use a laser. Now lasers are very, very nice for creating very small holes that we can burn through this mask. So let us imagine that I want to erase the mask at every probe location where I'm going to put the letter A, the nucleotide A specifically. Good point. Right, so I'm now going to burn a hole at only those probe locations that we're going to add the letter A. So the mask is intact except for those locations. Now I add a nucleotide A to be annealed to the surface, to be polymerized onto the surface. And only those spots where I burned through the mask get the letter A. Now I remask everything. Now I'm going to burn off all the locations where I want the letter C to be. And in some of those locations, it's the first letter to be added. But in other locations, I'm adding it to an A that was already there. So my laser comes across, and now I've burned through the mask at only those locations that I'm going to add the letter C. So we add the nucleotide C, it gets polymerized to whatever's there, and we again mask. And we keep doing this until we have probes that are a length of 25 nucleotides from the surface. It's the most bizarre thing, isn't it?
Mm-hmm.
Yes, question?
What are you using to mask? I don't understand.
Oh, I don't actually know what the chemistry is on this one, but the mask is basically a liquid layer that we add on top of this, and unless you burn through it, you're not going to be able to add anything to that location. So yeah, the mask is just it's a chemical alteration to all of the 3' ends of sequences that are available, preventing the addition of any nucleotide. It's a good question. I just don't know the chemical. Yes, sir?
The addition of the nucleotides, is that random? Or
No. No, the computer that is aiming the laser for us knows what sequences it needs to build. So it knows which ones are due for an A, and which ones are due for a C, which ones are due for a G, which ones are due for a T. And it keeps building and building until it has all of those built. Okay, so that's photolithography, and we can do it to produce these lengths of 25 nucleotides in length for these probe sequences. Now, how big is a gene? How big is a full transcript in comparison to 25 nucleotides? They're pretty big, right? Now, if you look at the gene in a human genome or something, you could be looking at megabases. But we're really talking about the messenger RNA derived from it, and that's actually much more compact. That's more like, say, a kilobase in length. So when we design the set of 25 mers we're going to measure for, we need to be very careful about picking 25 mers that are specific to a particular messenger RNA and that are in an accessible part of the messenger RNA as well. Okay, so there we go. This is the technology that was put together by Affymetrix for this purpose. And for a long time, they simply owned the microarray space, but I think that there's actually a lot more play in that market now. Okay, so at the bench, you're going to prepare or purchase a DNA microarray. If you have your own messenger RNAs you want to measure for, you'll probably use a spotted or printed array. But if you're just doing humans, for example, you'll probably just buy your microarray because there are already much better, more comprehensive microarrays available for us. Next, we're going to isolate a bunch of messenger RNA from these biological samples. Because messenger RNA is so twitchy, we're going to reverse transcribe it to produce a complementary DNA instead. We'll probably, if we're doing microarrays, we're likely to label those cDNAs by a dye. And that gives us the ability to measure these later by shining the appropriate wavelength on it to get light back. We hybridize the labeled cDNAs that we've created to our DNA microarray. And then we can wash off anything that's non-specifically bound and actually measure it by putting the appropriate light source across it and measuring the intensity that comes out. Now, I've put in this little note. This is always something that's worth talking about. Hybridization is what we're depending upon in using any microarray. Hybridization means that you have the complementary strands of sequence that are aligning to each other. Their sequences line up, A for T, C for G, etc. And we get hydrogen bonds forming between these pairs of nucleotide base, of nitrogen bases. So this formation of hydrogen bonds really only takes place if you've got perfectly specific sequences that can align and anneal perfectly. If you have mismatch bases in there, you know, one place where there's a G on this strand, there's a G on this strand, well, that sort of thing is really going to block annealing of the sequence to the other. So we always have to be, we should always be mindful that this hybridization process is the one we're depending upon for microarrays to tell us anything. Then we can ask, is the fluorescence we're receiving back from this microarray located at this probe or that probe, etc. So that's basically just a very high resolution camera at that point. Okay, now, in the old days, you would frequently see pictures of microarrays in which you saw green dots and red dots and yellow dots. So the reason you would see yellow dots is that in projected light, red light plus green light is yellow light. So when we saw a yellow color on these microarrays, it reflected the fact that both samples, the red labeled one and the green labeled one, were both emitting light at this particular probe sequence. Okay, now in today's methods, we don't typically combine two different samples on an individual microarray. And part of the reason for this is that frequently microarrays get used for experiments with a really large N, meaning that we have hundreds of patients that we want to profile for their RNA content. And as a result, it's a lot more straightforward for us if we can use each microarray on just one sample. It's also coincidentally very beneficial to microarray manufacturers who get to sell you one microarray for every sample you're going to do rather than letting you combine them. But that's neither here nor there. So we frequently will see that that different samples get separated to different microarrays. As a result, we have sort of a monochromatic single color analysis. Each microarray, we're simply asking, is this probe sequence, is this probe set, this particular set of complementary DNAs, are they bright or are they dark? If they're dark, we assume there's very little detection here. If it's nice and bright, that tells us we have a gene for which the expression is relatively high. Okay. Now, I've talked an awful lot about microarrays. It's very important, I think, though, that we stop for a moment and say, people who are trying to get really flexible and powerful senses of what's being transcribed in a cell do not limit themselves to microarrays. So let's talk about the number one weakness of microarrays. If you don't have a probe for it on a microarray, you can't see it, right? So if you happen to be looking at protein coding genes, there are lots and lots of microarrays out there that will let you detect the transcripts from every known protein coding gene. But what if you were also interested in transcription that takes place between protein coding genes in intergenic space? Something like a microarray is never going to have a probe for every location in intergenic space. So if your interest is to go beyond the protein coding genes, you're probably going to want to use something like an RNA Seq experiment instead. So it is possible to capture the 3' ends of messenger RNAs and sequence there. more commonly, in RNA-Seq style experiments, we will see people trying to capture sequence from all different patches across the transcripts. Okay? So, one of the most common things that we see in interpreting RNA-Seq data is a mapping process. So we know all of the messenger RNA transcripts that are projected for the human transcriptome. Therefore, if we sequence a bit of cDNA in an RNA-Seq experiment, we can figure out if it associates with this or that protein coding gene pretty straightforwardly. So this mapping process is one of the common ways that we go about making sense of our data. Sometimes we're not trying to summarize at the gene level to say this gene is producing a lot of transcripts. Instead, we may want to say which transcripts are dominating gene expression under these circumstances. In a case like that, we need to be paying special attention to sequencing reads that span 2 exons. Does everyone see that? Because if you're trying to figure out which exons have been brought together in the process of splicing, you need to prove that these 2 exons are brought together, you need to have a read that crosses from one into the other. So we frequently see that this mapping process needs to be isoform sensitive. Isoforms being a gene produces multiple isoforms when it produces multiple messenger RNAs. So we call each of those messenger RNAs an isoform in some cases. Okay, so what are the opportunities for us in moving to RNA sequencing rather than microarrays? Novel single nucleotide variants. Certainly, people care about detecting variant forms of genes that are being expressed. It's all fine and good to say this person is heterozygous at this allele, but it's a lot better if you're able to say not only is a minority allele present at this location, but we can also detect that it is being transcribed. Now you have not just an argument that this allele exists, but that it is also turned on in the process of gene expression. Fusions. Anyone hear of gene fusions? Okay, there are, I used to spend a lot of time in the cancer community, and frequently you find that cancer is associated with a lot of genomic rearrangement events. It can cause individual point mutations, but you can also find that regions of one chromosome get duplicated and inserted into altogether different chromosomes. And sometimes what you find is that one gene has now been spliced into another, that one transcript is being produced through 2 genes that used to be very far apart from each other in chromosomal structure. So if you're interested in gene fusions, you can detect that using an RNA-Seq experiment. It's a lot harder to do in the context of a microarray because, again, you must know what sequences you care about in advance and see that they're on your microarray if you're going to detect them by that method. So certainly people focus on fusions through RNA-Seq experiments. High dynamic range. So, as I mentioned, our ability to tell whether a gene is way up or way down is possible under both of these methods. One is using light from the fluorescent dye to make that measurement, and the other one is sequencing individual messenger RNA molecules, or cDNA molecules. However, we find that when there's a really big difference in intensity between very major genes, like say those for albumin, and those for vanishingly small genes, expressions, say those in a signaling pathway, that measurements by microarray are a little less reliable for us, that we can see over more orders of magnitude when we use messenger RNA sequencing, RNA-Seq, as our tool rather than microarrays. So dynamic range is another reason that we would shift to RNA-Seq. And finally, non-model organisms. Now I talked an awful lot about buying microarrays for human samples when you're doing large-scale clinical testing, but if you're dealing with platypus, how many commercial products do you think there are for buying microarrays in platypus? All right, extra credit if somebody ever finds one for me. But yeah, I think you're right. I'm not aware of anyone making microarrays for measuring gene expression in platypus. So if your model organism is non-model organism is the platypus, if that's what you actually care about, you're probably stuck with sequencing because you don't have an alternative to it. So you can do discovery in non-model organisms by this. All right. The other thing I was going to point to is just the very bottom part of this figure, which is to say we have an open reading frame for some organism. This is just one particular gene, one particular open reading frame for it. We have a whole spray of exonic reads, and as you can see, they're spread all over the entire length of the open reading frame here. As a result, we get a coverage map to say what nucleotides in our genome annotation for this gene are covered to what depth as we walk our way across this entire structure. So that's the kind of feedback you're going to get, a base resolved pattern of expression. How many different reads does this particular location in the genome correspond to? Now this gets kind of hairy as we start looking through what kinds of informatics are necessary for each of these structures. So reads throughout transcripts may be assembled to improve gene models or simply aligned to known annotation. Whew. If all you're trying to say is how much of this gene is being transcribed, you're in a pretty easy place for biotechnology to help you out. However, if you're trying to learn about mistakes in the human gene annotation, for example, you're going to need to be a lot subtler in how you make sense of your data. So let us start with the best case. In the best case, have I slipped off camera? Am I still doing okay? I'm good. All right, cool. So over here, we have a whole bunch of reads that we've produced across a gene. We can assemble these transcripts to recognize where these reads align with each other, and even align to a genome to say which exons map to which reads. This is what we were looking at in the last slide. So here we have an awful lot of information. We've got sequencing reads. They in themselves can be used to produce an assembly. But at base, we have a gene model that we can associate all of these with. That's really the best alternative. However, sometimes we're interested in putting together our first assessment of how the different exons seen for a gene can be stitched together to produce transcripts. So for something like this, you might know that you can assemble these sequences into contigs, and from that figure out which exons are being expressed. But you can also find information to help you know which splicing events are taking place, to know that these exons abut each other in a finished messenger RNA. So that's pretty good. Here we had to have an awful lot of information about all the transcripts that were possible from the gene. Here we didn't have that information. Sometimes we have very little information, and that would be like the non-model organism case. In this case, we may have no genome annotation to work from at all. One of the cases I've worked with in the Department of Biotechnology here is that of the chia, the superfood plant, basically. And chia doesn't have a published genome annotation yet. Somebody did go to the trouble in 2014 of publishing 5 different RNA Seq sets for chia, though, and we were able to use that to discern what the set of transcripts for chia could be. We put together a list of, I think, 56,000 transcripts that were observed in just these 5 samples processed by RNA Seq. So in some cases, you may be able to put together information about what are all the transcripts, even without having an annotation to work from. So in advance of the genome annotation being done, we can still get a sense of what transcripts are available from the species. You can't do stuff like that with microarrays at all. There's no microarray product that you can use from chia. Okay, so lots of flexibility and power in these data sets. I feel like I've already kind of talked about these, but it's valuable for us to think about the tool sets that allow us to do these things. The system I was using was called Trinity. Trinity uses this De Bruijn graphs that we talked about at the start of the month of April. So being able to create these graphs and de novo assemble transcripts out of just read information is possible. From that, you can try to figure out which ones you trust and put functional annotation on them. This is the process that we had to use for our CHIA project. In this case, we would be doing the microarray experiment, essentially, trying to figure out which genes are producing the most transcripts. That's kind of this middle path that we see. And sometimes you're trying to figure out whether you can trust the annotation that's been produced in the past for an organism. For that, this pathway over at the left is available. So there are lots of tools out there. Which one you use is going to depend on the purpose to which you're putting your data set. And just because someone produced a data set for one purpose does not mean that it can't be reapplied in one of these other contexts. All right. Now, there are an awful lot of different ways that you see people working with gene expression data. For most of the rest of this talk, we'll really be talking about the case where we have known transcripts for the organism and we're trying to figure out how much each of them is changing in response to some sort of biological difference. So we imagine biology as a cyclical process. Remember, we have a biological hypothesis. We create experiments that test that hypothesis. And from that, we refine that hypothesis to something new that reflects the new information we've gathered through those experiments. So you can see that although this slide starts at biological question, we see that after biological interpretation of this experiment, we generate new hypotheses, do a little bit of verification, and then we're right back into refining our biological questions. In the filled-in blobs here, you're seeing all the different places that bioinformatics tends to be very, very heavily involved. We're going to talk about a couple different means of data processing, such as normalization, Bland-Altman plots, things like that. And we'll talk about our strategies for how this data mining process takes place. The statistics are really going to start getting very involved once we talk about differential expression, but clustering and classification are things that we care about as well. And along the way, of course, there's the question of how do you store all that information, how do you integrate it together, and how do you visualize it? All right, so let's start with normalization. There are lots of reasons why you might observe systematic differences between an experiment run today and an experiment run, for example, next week. Some of these things can really interfere with your ability to compare the results from last week with the results from today. We would call that a batch effect. But for now, let's talk about normalization. We know that some systematic variation exists among our data sets. Rather than mislead ourselves into thinking these differences are biological in nature, it's appropriate for us to try to remove this systematic variation before we get to the place of biology. So, what about experimenter bias? Okay, you may think, well, bias is a terrible thing and no scientist should have bias. But we're human beings and we all have bias. So let me give you a very obvious example. I think each of you is going to do some sort of project as part of honors, in all likelihood. At the end of the day, if you compare two cohorts of samples, those that have been treated with magic drug X and those that have been treated with water, you're going to feel bias in the sense that you want something to be different. If you go through a 6-month project or a 6-year project for that matter, and come away with, well, these don't differ at all, you're going to be kind of disappointed, I think. Right? So you have a bias towards finding genes differential, and when you find those genes to be differential, you're going to convince yourself there's some sort of biological story that causes them to be differential. So we do carry biases into our experiment. It's not just bad investigators, it's all of us. We're human beings. We want to show our treatment is superior to whatever has been done before. We want to see that illness creates this particular set of genes to be differential that have this clear relationship. We all carry bias with us. All right, experimental conditions. What if we're having a fall like this one and we discover that the experiments for the controls are run during really balmy warm weather, and then the cases are all run the next week when it's suddenly rainy and dreary? Could it be that the environmental humidity conditions affected our experiments? We've observed cases in mass spectrometry where having painters working in the hallway outside caused our instruments to see different ions than if we didn't have painting going on outside. Biotechnology is really sensitive stuff. All right, sample preparation. Now, if you're a professor, it's almost an industrial practice that you learn to blame the person who was handling the pipette, right? Oh, the technician must have double pipetted these, or they pressed a little too hard and they got 15 rather than 10 microliters or whatever. This is not fair, but this is what happens. You, as the person holding the pipette at this point in your lives, probably would feel particularly sensitive about that, and for very good reason. So, it is true that occasionally we have minor variations that creep into our sample handling that we wish weren't there. It happens to everybody, and so being able to detect it from our data matters. And finally, machine parameters. It may be that, well, I'll just blame an undergrad. Why not? We all do. So maybe an undergrad wandered into the lab, and they were doing some experiment as part of some project they were doing, and they switched the program from 4A to 4B, which has some changes in the settings. Something like that may cause a systematic variation in your data. These things must be normalized away if we are to make use of the data. So in this case, we can use things like the metrics of the data we're producing to recognize them. So what could this look like? Let us imagine that we have run 8 microarrays. Okay? For each of these microarrays, we can ask what is the distribution of intensities for each gene. Right? So we have 22,000 genes being represented in each of these. We have this box that's showing us where most of the data live. Maybe this represents the middle 50%, the interquartile range. Have folks heard the term interquartile range? Mm-hmm. Cool, all right. So that means where do the data from 25th percentile to the 75th percentile live? This little box shows us that. And then we see that we have some values that are very, very high indeed. This is all on a log scale probably. So without normalization, we see that there are some slight differences among the 8 experiments that we ran. We might use something like global normalization. So in this strategy, we might ask, what is the median gene expression intensity that we have for all 22,000 genes for each of these 8 experiments? In global normalization, we've now multiplied each array by some constant to force either the median intensities or the averages to all be at the same level. So we see that these boxes do vary in how tall they are. But the midpoints have now all been forced to the same value. This normalization is one of these strategies that we would use. This is just global normalization. So this handles the fact that now all of these distributions are centered at the same point, but it hasn't done anything about how wide or narrow they are around that. You may instead use quantile normalization. And here the point is to make it so the 75th percentile for microarray 1 is located at the same place as 75th percentile for microarray 2 and 3 and 4. So you can see that now we have not only an alignment in where the medians appear, but in where the bounds of that distribution appear. So normalization is one of these techniques that we would use. You might ask, is it appropriate to use quantile normalization, or should I use global? If the cohorts differ in the breadth of expression profiles that they create, certainly using something like global normalization is going to be a lot safer for you than will global normalization. So which one is useful to you is going to depend upon how the data are distributed to begin with. All right. Now, there's also this MA plot concept, and this is one that's very, very useful in a broad variety of expression exercises. So you might have, imagine at the left, we have genes that have very sparse signals. We have very little light from them on the microarray, or we have very few reads from them in an RNA Seq experiment. It's really the same thing. We have a bunch of genes way down here at the low expression range. And we have a relatively small number of genes way up here at the high end. Those very, very intense genes are the ones that are expressed at really, really astoundingly high levels in the samples. Now, we might say, we should probably start with this hypothesis that kind of goes along with comparative experiments like this. Our hypothesis is that in a comparison between 2 cohorts, we anticipate most genes will not change. Some will. but most genes will not change. That's an assumption that we typically make just so our statistics actually work. So what we see then on this vertical axis is a log fold change. Something that's way up is something that's way up in one cohort and way down in the other. That situation could be reversed, that it's high in B and low in A, which brings it way down here. But most genes fall right along the midline. So in an MA plot, we're hoping to see a nice spread from left to right, where we don't see a big bias towards being up in B and low in A, or up in A and low in B, that's characteristic of a particular abundance. So the MA plot lets us check for bias as a function of abundance. Everyone sees that? Okay. Yes. All right. Lots of people make use of biclustering as well. Biclustering is probably one of the most characteristic visualizations that we ever see in differential gene expression. So why do we do this? Imagine two sets of relationships. The first relationship is that we have all these different people that we did microarrays on or that we did RNA Seq upon. Some of these people have very similar gene expression profiles. Some have very dissimilar gene expression profiles. So you can cluster together people based on how similar their gene expressions are. Everyone sees that? Okay. So if you were to do this with a data set where you knew you had only 2 cohorts, you would probably hope that all of your cases clustered together and all of your controls clustered together. If you have just 2 cohorts and you find that their clustering profiles are all intermixed, that's not a great sign for you, right? It means that the comparison you're going to try to do is going to be have a lot of commingled effects. So that would be rough. So you might find that you, in this case, could find certain cell lines have similar gene expression profiles. That's what each of these columns represents. So you might imagine, for example, that hepatic cancers, liver cancers, would all group together, and brain cancers would group together, and breast cancers would group together. Something like that would be your expected result if you do a bunch of cancer cell lines. Next, each of these microarrays is measuring a whole lot of genes, say 20,000. Some of those genes are going to have a lot of missingness, meaning that you had no measurement of expression for those genes in several of these samples. So you might throw those out. Maybe now you're down to 10,000 genes that had enough observations. Next, you would say, well, many of these genes show very little variance among all of the samples. Well, something like that's not going to be very informative. You're not going to find a difference for genes that have very little variance. So you might next say, I must have a coefficient of variation of at least 10% if I am to include this gene in further analysis. So now you're down to genes that were observed across all the samples and that had pretty good levels of intensity. And you can then ask which genes are most similar in their expression patterns across the cell lines. So across the X here, we've grouped together samples that are most similar in gene expression. And on the Y-axis, we've grouped together genes that are most similar in their patterns of expression across cell lines. You see, so we're clustering on the X-axis and clustering on the Y-axis. And that gives us what's called the checkerboard. Now, if you're a male in this room, you may see very little distinction between the red and the greens. If so, color blindness is difficult for red-green plots. We really could have chosen a better combination of colors than red and green for these plots. Correspondingly, you will see people doing blue versus yellow plots for these. Those options are good ones for you. So, if you're not colorblind, hopefully you're seeing red here, red there, green up top, and green over here. Grand. So, is anyone not seeing differences in those colors? I'm just curious. I've been fortunate that I have color vision, but I know not everyone does. All right, so this checkerboard pattern is one of the ways that we're able to say, this set of cell lines at the left differ from these others that we see at the right in red and in yellow in this set of genes or in that set of genes. Not always will it be that clear cut. Many genes will have flecks of red buried in with the green and so on. But ideally, biclustering is very useful for you to do discovery on your data sets and discover which genes are most similar across samples and which samples are most similar in gene expression. Am I belaboring that too much? Okay, we use those figures all the time, so I felt that it was useful to kind of walk through it a little pedantically. I hope that's useful. The Volcano Plot is another example of a visualization that people use all the time in differential gene expression. So we're going to spend a lot of time talking about p-values in just a moment, but for now, I want you to just evaluate that a low p-value for us is the suggestion that this gene is differential between two cohorts. All right, this is a hit for us. Now, on the Volcano Plot, we use the p-values, but not just as they're produced. We're using negative log base 10 of the p-value. If you remember my talking about Fred a while back, Fred scores, remember we expressed the probability of error for an individual nucleotide by taking its negative log and multiplying by 10. The same can be done in this context. So if you have a high value, that is to say it's a very low p-value. If you have a low, if you show up very low here, it means your p-value is correspondingly high. So it's kind of an inverted relationship. The whole point of this is to get the hits way up high, the differential genes way up high, and the other stuff way down low. That's the y-axis. Now on the x-axis, we have a polarity. Remember, to say that a gene is differential is cool, but you'd really rather know, in addition, how much it changes, which direction it changes. So it could be that a gene is differential because it's way high in A and way low in B. Or it could be that it's way low in A and way high in B. Those are very different effects. Imagine that you were trying to make an argument that a certain pathway is way up under a certain circumstance. It would be embarrassing if multiple genes within that pathway had p-values, but their directions were all opposite each other, right? To have 4 genes from this pathway be way down and another 4 genes from this pathway be way up. You would need to have a little more subtlety in how you interpreted something like that. So we want to know not only that the gene is differential and thus high, but to express the direction of that difference. So in effect, what we have is an axis sitting right here at log fold change 0. So log base 2 of fold change equal to 0. What does that mean? Okay, great. So let us say that I have a value of 1,000 on average for this gene in cohort A, and I have a value averaging 1,000 for this gene in B. What's the fold change here? 1,000 divided by 1,000 is 1. Great. So that is a 1-to-1 fold change of 1. When we look at that on the log scale, we're asking, to what power do I need to raise 2 to get this fold change? So 2 needs to be raised to what value for us to get the value 1?
0.
0. Very nice. Great. Good catch. Okay, so when we have a log base whatever fold change of 0, that means the values are equal. If you have a log base 2 fold change of -1, it means that you had, say, a 500 versus 1,000. Right? This is 1/2 of this other value. And if you balance it the other way, say 2,000 to 1,000, you have a log base 2 fold change of +1. So this fold change is just as extreme as this fold change, but they're in opposite directions. Okay, so I'm going to just give you a bit of biological lore here. In general, when people interpret these, they say, I'm not interested in any fold change less than 2-fold. Okay? So on a log base 2 fold change map, that means we're going to draw on a line right here at 1, because that means twofold change up. And we're going to draw in another vertical line at -1, meaning twofold change down. All right, so just imagine, whack, we've thrown these barriers in here. Right away, the biologist has said, I don't care about anything in the middle here. Those changes aren't extreme enough that they're biologically important. This is a very common way to read these, so I want you to know about it. Next, I don't have a stick in here, do I? I always miss having a good stick when I'm having a lecture. It's fine. So let us imagine now that we're going to draw a horizontal line across this plot. This horizontal line is going to say everything above this is a hit, everything below this is a miss, is not differential. So we're making one criterion on the basis of log base twofold change. These goalposts right here. Do you have goalposts in rugby? You have goalposts in rugby, right? Right, okay. So imagine these are goalposts, right? Something has to be outside these goalposts to be a big enough change for us to care about. And it needs to be above, right there, that line, to be a hit, to be a significant p-value. So in effect, in the volcano plot, we're trying to draw attention to the genes that fall in this quadrant right here, which is to say significant p-values and very, very low down. And we want to look at things in this quadrant right here. Significant p-values being that they're high and way up in the other cohort. And the rest of this diagram, we just use it to say, well, that's what the data look like. These are the ones we're not paying attention to. So in effect, this one graph gets decomposed into 6 sectors, and the only 2 we care about are things that are way down and significant and things that are way up and significant. All right. Volcano plot. Why do we call it a volcano plot? Exactly. That was just, it's the perfect answer right there. So yes, we imagine that we have a little Mount Vesuvius blowing up here, and we've got genes spilling out of it. It's a rare evocative name. I mean, it's a better name than MA plot, isn't it? Yeah. Right. Okay. Now, one could give many, many lectures about machine learning. Some of you, I think, may have signed up for a special topic in biomarkers later in the year. I'm really looking forward to that because I'll be giving a lecture on machine learning in that segment. It's my first time trying to teach that subject, so bear with me. It's going to be fun. But how do we go after stuff like this? What would machine learning be useful for? I'm going to try to walk you through 3 different applications of machine learning that would be very valuable for us. So first off, class comparison. What genes differentiate multiple cohorts? So let me say that I have a bunch of microarrays for folks who have cancer and a bunch of microarrays for folks who don't have cancer. And I want to know which genes are most differentiating between folks who do have cancer and folks who don't. Okay, so this is called a differential expression analysis. That's all great. Our input is not only the gene expression data, but a kind of metadata. Metadata, that's a term we didn't talk about here. I talk about it a whole lot in the 8 days stuff. So we have not just the microarrays, but we have a class label on each one. So for each microarray data set we produce for our differential expression analysis, we're going to indicate this is a cancer or this isn't a cancer. And from that, we're going to get a list of genes that are the best ones to tell us which genes are those that differ the most between these 2 cohorts. All right, so that's kind of the most commonplace kind of analysis that we do in differential gene expression. You might be interested in class detection, though. How many diseases is breast cancer? How many diseases is breast cancer? If you get breast cancer, do you have the same thing? Do we treat all breast cancers the same way? We do not. Now, that's a change, right? At one point, we thought, well, if you have breast cancer, you have breast cancer. Why would you differentiate? In fact, you can use the data like those from microarrays to ask about what causes one breast cancer to look very different from another. So, if you have microarrays from a bunch of people with breast cancer, you can use biclustering to align together those folks who have cancers with the most similar gene expressions. And from that, you can see that certain subtypes, such as triple-negative breast cancer, have people heard of this? Triple-negative breast cancer? It's one of the nastiest kinds to get, basically. It means that three of the genes that are all responsible for this cancer becoming faster, growing faster, and getting more adventurous throughout the body have been knocked out. So triple-negative is a bad diagnosis as breast cancers go. So we learned about that by using clustering analysis, where we gave the software all of the gene expression data and asked it to tell us which samples were most similar to each other. There we saw that we were getting segregation among the different sets, the different types of breast cancers. Okay, so the output is a group of similar samples or similar genes. This is the bi-clustering that we talked about. Class prediction, which genes are predictive of outcome? This is much closer to the biomarker question. And if you're interested in biomarkers, I definitely suggest you take that special topic. The gal who's teaching that, Dr. Caroline Beltran, is amazing in this subject. And I think she's going to have really cool things to say about it. So for this, machine learning almost always ends up getting called into effect. We must split our data into a training set and a test set. So we're going to have to teach our software how to find examples of X with our training set, and then the output will give us a prediction model that we can then test in our test set to say how frequently did it get the label right on these other samples. Lots more to say about that subject. Okay, now let's talk about statistics. What time are we sitting at? Am I running on too long?
20 past 11.
20 past 11, okay. We're still going to get out of here easily by noon. Anyone have a lunch date at noon? Anyone have a lunch date before noon? Okay, good. All right, I'm not going to feel I'm wasting your time then. This is my last chance to get to talk to everybody, so. All right. Let us imagine that we're doing a differential expression analysis. I have a bunch of cases, and I have a bunch of controls, and I have some set of genes. What are these identifiers we see over at the left? Like 1405 underscore I underscore AT. These are probe names, essentially. So these are particular probes that we have on a microarray, and we're associating those with some amount of intensity from microarrays. So we have probes, those are mappable to genes, and then for each gene we have some level of expression that we've seen. We have a null hypothesis. If the term null hypothesis is one you've never heard before, I would strongly suggest that you spend some time with the statistically speaking lectures that you received links to. Because statistics is something that just is always going to be part of biotechnology. You can't be a great biotechnologist with no exposure to statistics, I'm sorry to say. So spend some time there if you need to. The null hypothesis that most typically gets tested as part of differential gene expression is to say, everyone saw this character, everyone knows what that letter is? What language does this letter come from? Greek. That is a Greek letter mu. All right? I don't know if you've encountered this, but when you're in Greece, if you see cows on the hillside, they don't say mu. They say mu. It's true. All right, so here we see it. We have a bunch of intensity values. We have an average value, and just as shorthand, we use the mu to say this is the average value seen in this cohort, and this is the average value seen in this cohort for a particular gene. So I have a hypothesis for each row, and for each row I'm going to test is the average for the A's the same as the average for the B's. All right. The alternative hypothesis is just everything else. What if the means are not the same? In that case, we have an alternative hypothesis to explain what we've seen, which is to say, well, the means are not the same. So if you're using something like t-test, which is widely used, even when it's not appropriate, we're evaluating the probability that the observed sample means would be as different as this by random chance alone. Okay, so that is a little bit of background. So for each gene, we have a different statistical test. We're going to use the t-test in all of them, let's say. But for each one, we're going to take each row of intensity values separately from all the others. So t-test doesn't get to know the results for gene 1000 when we're doing gene 999. Each one is entirely handled entirely separately from each other. Okay, now, why would we use t-test or u-test? U-test is a pretty common one that we might use. They're available in GraphPad Prism, or they're available in Excel and so on. But let us imagine one gene under very slightly different circumstances. Under this top case, gene X got 9.61, 11.03, 10.5. And below we see 9.61, 11.03, 10.5, right? So the expression in cohort A was identical for these. On the right, though, we see that we have 11.44, 12.23, and 13.61. That's different than what we saw in the B cohort in that one value. See that last one? It jumped from 13.61 to 25.61. Right? That's a huge jump. How does that affect the p-value that comes out of these two statistics? If we do a t-test, we see that the first one has a p-value of 0.06. By changing only one value, our p-value jumped to p of 0.32. So just one value changed within the expressions, but that change was enough to boost variance, and thus the p-value rose, taking it from something that looked like it was going to be significant but wasn't quite, to something that's clearly not a significant difference. All right, what happened to u-test under these same circumstances? U-test gave exactly the same p-value in both cases. How could two statistical tests come up with such different ideas of what changed or didn't? The reason is that t-test cares about magnitudes. So 13.61 was a high value, and we like that. We're kind of making the assertion here that B is higher than A, right? You've got this 11.44, 12.23, 13.61. Those are all higher than the three values we saw in the A cohort in magenta here. So how is it that by making one of these values even higher than that made things worse? How did that compromise a p-value? The answer, and this is something I repeat a lot in the statistically speaking course, t-test and u-test don't just care that things are different. They care about the variability of difference as well. So if you're looking for a difference, you need not only to have a difference between the mean values, but you need to have low variance about those means as well. Okay, so in this case, we see that the p-value from t-test gets a lot worse by changing one value because it's broadening the variance of the blue cohort versus the magenta. Okay, the next thing is that t-test and u-test differ from each other significantly. Quite a lot in any case, because the u-test doesn't care about magnitudes. It only cares about orderings. Okay? So in, let's say, the top vector, what's the highest value we see? 13.61. All right, so we can write, draw in your book a top ball, let's say, and it has the number 13.61 on it, but really all we care about is its rank. Its rank is number 1, it's the highest value. What's the second highest value we have? 12.23, so you can write in your notes another ball below that first one. It has a rank of 2, it has a label on it, 12.23. All right, now if you keep going down that line, you're going to see that you have 3 blue balls at the top and 3 magenta balls below that. To u-test, this is ideal. U-test only cares that all the balls of one color are above all the balls of the other color. You'll note, though, that it didn't give it a statistically significant p-value. It doesn't have a value below 0.05. And the reason is that u-test requires that you have a larger number of samples to work with, right? So if you had 5 blue balls above 5 magenta balls, the u-test is going to give you a much lower p-value. And the reason for this is that there are more ways that you can interdigitate 10 balls than you can 6. Okay, so one of the things that I hope you take away from that is that when you're designing your experiments, you know you can get a lot more statistical power if you have 5 replicates of each cohort than if you have only 3. Keep that in the back of your mind. I'm just going to point this out for a moment. Professors, we're cheap, right? We have limited amounts of funds for stuff, and we try to get by with the smallest experiment we possibly can. As a result, at some point in your career, you're going to be pressured to do an experiment with a 3-on-3 comparison without knowing whether that's got enough power to give the answer you want. If you always argue for 5 replicates rather than 3, there's a lot more times when you're going to be able to discover differences just because you have more power in a 5-on-5. All right, I've said that. All right, so now we've talked a little bit about the differences where t-test and u-test may be used for difference testing. So this is not the only set of options that we've got, though. If you're going to deal with a fairly large and complex data set, you probably want linear models for microarray data, lemma. Now this is a tool that's not available in Excel. This is an R tool. R is one of the most rapidly growing tool sets that we use for biostatistics today. It has all kinds of software available for dealing with almost any imaginable kind of data analysis. It's your best friend. But R is a little choosy about who its best friends are, right? So becoming proficient at R probably requires a bit of practice from you. It is not an easy thing to get on top of, but there are people here who can help you do that. There are lots of people up in Sanby, for example, using R. I can teach you some of the basics of R. So I hope that you'll find that useful. Okay, so lemma is one of these libraries available for R, and it includes something called an empirical Bayes, or moderated t-statistic test. One of the problems that we frequently encounter with dealing with microarray data in the context of t-test and u-test is that our ability to estimate variance is very weak when we have 3 or just 5 samples. So the moderated t-test helps it to give us a much broader assessment of how much variability can be found in our data. And as a result, the lemma version of t-test with this moderated t-statistic is an awful lot better at finding truly significant genes as opposed to those that are much more marginal. Okay, so very good to know that there are better power tools available to help you. To return to this graph, I note that each row is a test. How many genes do we have in the genome? 20,000. If you do 20,000 t-tests of data that have no differences in them whatsoever, what fraction of them should have a p-value below 0.05? If there's no real difference between cohorts, what fraction of t-tests should produce a p-value less than 0.05? Okay, no one knows this one. So I'll just explain that when there are no differences between cohorts, if we're dealing with totally random data, we expect a uniform distribution of p-values. So what that is to say is that if you do 1,000 t-tests, 1,000 t-tests where there are no real differences, 5% of them should produce p-values below 0.05. So if you do 1,000 t-tests, how many of them are producing hits? 50, very nice. Yes, 5% of 1,000 is 50. So if you were just testing 1,000 probes with random data, 50 of them should show up as hits, even when there's no real differences between the cohorts. That is bad news for us. And that means we need multiple testing correction. Now, multiple testing correction is a fairly complex topic, but there are a couple things, a couple terms that I would expect everybody to understand about it. The first of these is Bonferroni Correction. Bonferroni is, I believe, an Italian mathematician from way back in the day. He was never involved with multiple testing correction, but he created something called the Bonferroni inequalities that a biostatistician from America, a woman actually, discovered could be usable for asking how problematic these multiple tests would be. So let us imagine that we have done 1,000 t-tests, right? We just set up the scenario, 1,000 t-tests in the context of random data. she says that you can use Bonferroni's inequalities to say, I'm doing 1,000 t-tests, therefore my number of trials is 1,000. Okay? And as a result, she says, instead of looking for a p-value that's less than 0.05, you should instead look for a p-value that's less than 0.05 divided by 1,000. Whoa! Right? That's quite the correction, isn't it? Suddenly, all your hits go away. This is the number one thing people complain about in handling multiple testing correction. So, her argument is that because you're doing many t-tests, you want to protect yourself against making even one claim of a differential gene when there are none differential. This is a very heavy claim, right? She's saying that if you claim, if you're claiming some set of genes, you're trying as differential, you're trying to protect yourself against claiming even one that's incorrect. Claiming a gene is differential when it's not. She wants to protect you against making any false claims for that set. And the Bonferroni correction is thus one of the, is a very conservative kind of protection against multiple tests. Instead, you'll frequently find that people use the Benjamini-Hochberg false discovery rate estimation instead. So remember, Bonferroni is going to try to protect us against making any false claims of difference. Benjamini-Hochberg is going to try to limit the rate at which we make false claims. Okay? So let's compare these scenarios. In 1,000 t-tests, if I want to protect myself against making any false claims, I need to shove my p-value threshold down by 1,000-fold. That's the Bonferroni approach. With FDR, I might instead say, I want to keep claiming genes until I've reached a level at which 5% of them may be estimated to be erroneous. It's not going to tell you which ones are erroneous. It's going to tell you that the fraction of these false claims that I'm willing to tolerate as errors is 5%. So this Benjamini-Hochberg approach ends up being very popular as a result. So you're going to start by sorting all of the genes by the p-values that they produced. So if you had 1,000 genes you are measuring, you've got 1,000 p-values, and you just sort them from lowest to highest. So each of these little pips represents a gene's p-value. Next, you have an escalating threshold. You see how this red line slopes gradually upward? You can compute the threshold to be applied to the first p-value here, the second p-value here, and the third p-value here. So it's a linear rise in the threshold you apply. So when you find that your data have crossed over this line, you draw in your limit and say, okay, this is the set I'm going to claim as my differential genes. So that's pretty cool. Now we can say, these are the sets of hits, but we are cognizant of the fact some fraction of them are false positives. That's tolerable. Now the statistician isn't going to cream you when you send your paper in for publication. Now they're going to say, they were aware of multiple testing correction, they incorporated it into their findings, and the results are therefore not something I'm going to reject out of hand. It's embarrassing to send a paper in for publication and have a statistician rip it apart. Trust me, been there. So, multiple testing correction is one of these things you'd better know about because we're very, very frequently in biotechnology measuring lots of things. Lots of genes, lots of lipids, whatever. And this concept of multiple testing is not just about microarrays. Multiple testing is throughout biotechnology. So this is something you'd better know about. Okay, now, where do we run afoul of things in multiple testing? Let us start with all my hits vanished after multiple testing correction. This is the number one thing professors come to me complaining about. You might think it's just grad students. It's not. Professors come to me saying, all of my hits went away when we used multiple testing correction. Our field is very slowly becoming more statistically literate. Sometimes the null distribution is correct. The null hypothesis claims that there's no difference between these cohorts. Sometimes, biologically, that's true. Your professor thought, perhaps, that treating them with drug X rather than drug Y would cause these genes to show up differently. They didn't. Sometimes the null hypothesis is for real. Sometimes there is no difference to be measured. Maybe your experiment was underpowered. Power is a really big word in biostatistics. People use it all the time. It's actually kind of a difficult concept to teach. But maybe, in order for you to find that gene to be differential, you needed to do a 7-on-7 comparison, not a 3-on-3 comparison. If your experiment was underpowered, maybe this gene could have been a hit, but wasn't, because your experiment wasn't big enough. All right? So sometimes these hits that people are complaining about vanishing aren't really hits at all. Sometimes their experiment was the wrong one to detect it. Next up. This is one that really chafes my hide. It makes me very, very angry indeed. Dr. X published without using multiple testing correction, so why must I? I think you all have encountered some name that shows up in the literature you're reading for class and stuff. There's some professor who's big and wealthy and has amazing labs and has a continuing flow of postdocs and people heading off to be professors themselves. And you think to yourself, my goodness, how does this professor do it? Sometimes we discover that these big hat professors are not actually very good statistically. And if we start looking at their data much more closely, we discover that their claims of detecting X, Y, or Z, that these are not in fact real when we look at the underlying data. And we may occasionally discover that statistical controls that should have been placed in their research were not. However, there is a double standard at play. Because Professor X has been publishing papers for 20 years. As a result, when they send out a paper for review, people assume that they did the right thing, even if they didn't. You, as a new professor, or as a new postdoc, or a PhD student, sending your paper out for review, you get it looked over with minute detail by somebody who's just beating you up in their comments on your manuscript. That is because you're new in the field. And they don't assume, they don't make the same presumption that you're doing things right that they make of Professor X. This is a double standard and it's not cool, but it happens, so beware. When you publish your papers, you need to be much, much more careful than I do. I'm going to get away with crap that you don't. When you've been in the field for 20 years, hopefully people will cut you the same slack that they cut me now. So, as a grad student, you're going to be placed under more scrutiny as you push papers forward. Dr. X is getting away with bad practice. We don't like this. So, don't be one of the people who's publishing using bad statistics. All right, next. Sometimes people do the wrong test in the wrong setting.
We were going to be here, but we'll move. It's fine.
I think we're till noon.
The booking sheet said that we were here at 11:30, but don't worry.
Okay, we'll be.
We'll move.
We're shutting down pretty fast.
Okay.
Yep. Imagine that all you know how to do is a t-test. Is that far wrong? I mean, some people here are statisticians probably, but some of you feel comfortable doing t-tests and nothing else. And so when you're given an experimental design where you have cell cultures of types A, B, C, D, and E, and you want to know where the differences lie among them, you might fall into the trap of comparing A to B, and A to C, and A to D, and A to E, and then B to C, and B to D, and B to E, and then C to D, and then C to E, and then D to E. Done, right? It's not actually okay. Not actually okay. In a case like that, you've now done 10 pairwise comparisons. And just by doing that, even on a single value, you already need multiple testing correction. So doing these tests properly by something like a Tukey honestly significant difference test would be a whole lot safer for you because it's already got multiple testing built into it. So even in a case like the one I described, where you're only measuring one value across 5 cultures, you can still get yourself into trouble with multiple testing correction. I want to bring the advice of Ronald Coase to bear here. If you torture the data long enough, it will confess. If you remember one thing from me, I hope it would be this. Science is not the process of producing a significant p-value. It's not. At some point in your lives, you're going to have some experiment you're analyzing that you wanted to have a difference and you didn't get one. And the goal is not to keep trying different statistical tests until one of them gives you a p-value below 0.05. Be careful. So work hard on having defensible statistical strategies. Do not torture your data until something you want to produce a value of below 0.05 shows up. Everyone will feel happier for it. So our takeaway message is, hybridization-based assays by microarray are rapidly ceding ground now to high-throughput sequencing methods, things like RNA Seq. It's been cleaning the clock of microarrays for most research purposes. That said, microarrays are still a much less expensive, much easier analyzed way to get at gene expression. So just because RNA Seq is available does not mean it makes financially the best sense. High-throughput sequencing data places considerably more load on bioinformatics for interpretation than does Sanger sequencing, or than does microarrays for that matter. But the data are far richer, and the kinds of questions you can ask with them are much more interesting ones. Plots to visualize data quality and key measures of performance are vital to understanding complex issues that can crop up in these experiments. Looking at normalization plots, looking at MA plots to evaluate bias as a function of abundance, these are all quite essential. Systems biology can easily yield situations where multiple testing is a problem. Bonferroni and Benjamini-Hochberg FDR really matter. They can make your life a lot better. They can protect you from that evil person who is reviewer 3. So I hope that this was useful to you. I've really enjoyed talking with you all. It's been a lot of fun. As I've said, if you're taking this special topic in biomarkers, I'll see you again. But for now, just know that you can come visit me on Thursdays over at the other end of the department. I'd be glad to talk with you and help you figure out how to make the best sense of your data. Thank you very much, everybody.
Thank you.
Thank you.